Hi, Jeff. It's Christina.
Hi.
Christina.
I've invited Fu here to also participate.
Great.
Can you hear us okay?
Yes.
Awesome.
Okay, well, it's straight up noon, and so I'd like to go ahead and get started. My name is Jeff Obey. I'm a professor at the University of North Carolina, and this year I'm the chair of the Division of Organic Chemistry of the American Chemical Society. And so I'd like to welcome everybody to today's virtual symposium, which has been ongoing in the division for about a year. And so, as you can see, we have two talks today, both tilting towards chemical biology with a strong carbohydrate flavor, I suspect. One by a new investigator who is off to a great start, and another by a very distinguished investigator in the prime of her scientific career. So here are some links on the page that you can see for the symposia, which can provide one at the bottom that I want to pay particular attention, because that's where you can ask questions if you would like. During the symposium, what I'll be doing is I will be taking questions down, and if time permits, be conveying them to the speakers. These videos are archived, as are many other videos, which you can see. I'm trying to keep these slides wrangled here. Which you can see on the URL here, and certainly I'd recommend that you go there, including a lot of the talks from the National Organic Symposium, which was held this last summer in Indiana, at Indiana University at Bloomington. The next NOS will be in 2 years at La Jolla. We'd like to thank our friends at the Center for Selective CH Functionalization at Emory University for their help in putting this together. We have piggybacked on their very nice video conferencing capabilities, having nothing to do with my inability to control the slides here, but they're one of their upcoming meetings. I'd also like to specifically thank Rachel Hall from Emory, who has helped us with this. And our own symposium is led by Carl Hansen from Boston Pharmaceuticals, who's really put together this program over the last year. Just a little bit of news from the DOC. This year we had the LEAD Award awarded. It's every 2 years, and it honors excellence in teaching and research. It went to Neil Garg at UCLA, so congratulations to Neil. In addition, a number of our colleagues were selected as fellows to the American Chemical Society, and the names of division members who have received that honor are shown on that recent slide. Now, I'd also like to mention that we are in the waning days of a slogan contest. We're looking for a slogan for the Division of Organic Chemistry. The entries are due within the week, as you can see, by October 7th. And the winner will receive registration to ACS National Meeting and an invitation to our award dinner. So get those entries in if you would. Of course, we're already getting ready for and looking forward to the Philadelphia National Meeting coming up in March. And there will be a full program, as always, at ACS in 2020. You can see some of the symposia that will be held at that meeting, and here's some more. Of course, one of the attributes of an ACS meeting is that it includes a number of award talks. And so some of the awardees that we will be honoring at that meeting are shown here on this slide. And so congratulations to all of them, and certainly we're looking forward to a number of great symposia for that on that occasion. If you're watching, and hopefully we have a lot of groups watching from around the world, especially if you're in a watch party, tweet us a picture. We'd love to see who you are and where you are. And so here's the Twitter handle for the division. And we try to use Twitter as much as possible for the power of good and in promoting the division and letting our members know what's going on. I'd also like to mention that we support scientific programming and symposia aside from those at ACS meeting, and those, if you're interested in working at the seeking DOC support for your symposium, you can do so by going to this website and making an application. So finally, we hope that most of you watching are members, but those of you who aren't, we certainly welcome you to join us, and you can go to organicdivision.org for information as to how to do that, and specifically use this link to actually become a member. Okay, so let me see if I can now stop sharing those slides. And I apologize if anybody got whiplash from my going back and forth on the slides there. There we go. Okay, so it is now my pleasure to introduce our first speaker. This is Professor Christina Wu. Professor Wu is a faculty member at Harvard University. She received her PhD from Yale in 2013, working for Professor Seth Herzon as an NSF predoctoral fellow. She then joined the laboratory of our second speaker, Carolyn Bertozzi, at Berkeley as a postdoctoral fellow, and then moved on to Stanford as that group moved. And she's been a faculty member at Harvard since 2016. In that time, she's already been recognized by a number of awards, and I'll just mention the Alfred P. Sloan Research Fellowship and Bayer Early Excellence in Science Award. So without further ado, turn it over to Professor Wu.
Thank you, Jeff, for that kind introduction. And I also wanted to introduce Wu here, Cha-Fu Chang, who's a postdoctoral fellow in my group. He is going to present some work to everybody today that will be the first time that it's seeing anything outside of my group. So I'm quite excited to share this with everybody today, and as is Fu. And Fu did his graduate work with Richard Taylor at Notre Dame University, and has been working with me for, I guess, 2 years or so. Yeah. So we're super excited to be able to share our work with everybody today, especially given our organic roots and chemical biology leaning. So I'll specifically describe the work that we've been doing towards binding site hotspot mapping and how we've been using this to discover new strategies for characterizing small molecule protein interactions. So my lab is specifically interested in understanding how small molecules regulate the proteome. And we characterize this from the perspective that nature has learned how to do this a lot better than we have. And this is certainly a perspective that I gained and learned while I was in Carolyn's lab, as well as in my own training in Seth's lab as well. And so one of the primary foci for us is trying to understand how small molecules like glycans are regulating proteins, as well as other post-translational modifications such as phosphorylation and ubiquitinylation, such as there is quite a bit of crosstalk between these different post-translational modifications. Our goal here is to ultimately reveal a significant number of functions for these glycan-based post-translational modifications, while simultaneously characterizing how small molecules can interface with proteins. So our main goal in this aspect of our research, for which Fu and most of the work I'll talk about today is geared towards, is towards trying to escape, say, just kind of hitting enzyme binding pockets and being able to target and characterize the interaction of additional ligands binding sites of small molecules. and ultimately to reveal how small molecules can regulate protein-protein interactions, with the overarching idea being that as we can discover how these small molecules are interacting with proteins, hopefully eventually this work will feed back on understanding the functions of glycan-based post-translational modifications, particularly as many of these kinds of modifications are much more, or interactions are much more similar to how other nature-based post-translational modifications are behaving. And one of the ways that we've been kind of thinking about and motivating the work on our glycobiology aspect has started to come and leaked back to how I think a lot of the chemical biology field has been thinking about protein degradation. And so here, the work that we've been starting to think about is specifically geared towards the fact that there's right now a huge impetus in the field of chemical biology towards learning how to alter and regulate the E3 ligase or ubiquitin proteasome to attack and target new neosubstrates through both inspired by initial immunomodulatory drugs, but now more work by PROTACs and targeted protein degradation. And ultimately what this is essentially resurfacing or altering the ubiquitin proteasome. And so we started to think about how we can do that for glycosylation and specifically develop small molecule chemical adapters of glycosylation, such as for O-glycan transferase, introducing a glycan, or for O-glycanase, removing that glycan using these small molecules. And so how can we identify small molecules like this that can mediate or ligand interactions between two proteins? And that's what I'll primarily talk about today. And our idea here is that ultimately, as we can understand the glycosylation functions, we can understand how to stabilize or destabilize these proteins more broadly. So when we began to think about how we might be able to reveal those interactions, we became particularly interested and focused on photoaffinity labeling chemistry. And the kinds of photoaffinity labeling functional groups you have on hand are shown here, including diazoacetic acid, the one first used by Frank Westheimer, actually at Harvard. And now more recent functional groups like the benzophenone, phenylazide, and the diazurine. And what happens when you take these functional groups, for example, the diazurine, and you photolyze them, they generate a reactive intermediate that can sit in either a singlet or a triplet fashion. And this reactive intermediate then rapidly inserts into protein substrate. And so the idea here was that if we can readily characterize how small molecules are interacting with proteins using photoaffinity labeling, potentially we can use that to discover how small molecules can mediate protein-protein interactions and maybe control post-translational modifications. And so when Frank Westheimer, who initiated the field of photoaffinity labeling, was looking back on the work that he had done, he kind of summarized this work in this table of desirable properties for reagents for photoaffinity labeling. And these properties make sense, more or less. The desirable photoaffinity labeling functional groups should be stable to heat and acid until you want them to be photolysed, and they should be photolysed rapidly and at wavelengths that don't impact the cell. As well as, and most importantly for photoaffinity labeling chemistry, this insertion event should be rapid and indiscriminate for the insertion into a protein intermediate. And this was primarily what attracted us and many others to the field of photoaffinity labeling, that this RH group could be one of many different functional groups found within the proteome. And that this kind of insertion would enable us to identify a broad range of binding interactions of small molecules. And finally, Frank Westheimer pointed out this last point here, which was that there should be little or no rearrangement of that photoaffinity labeling functional group. And it was this kind of last point that he led to a concept that he called pseudo-photoaffinity labeling. In other words, photoaffinity labeling chemistry that occurred not through the diazurine or through this carbene intermediate, but through some other intermediate. And we felt that this had subsequently been lost from some of the discussion in the literature. So we're not the first to be interested in chemical proteomics and particularly photoaffinity labeling. Many of the other key players are shown here, particularly in activity-based protein profiling from Vancouver and coworkers, and now expansion to being able to characterize the ligandable proteome. But what seemed to us to be a major challenge as the field shifted from, say, looking at covalent chemistries for targeting proteins towards now photoaffinity labeling chemistries was the fact that when you take a small molecule and you say derivatize it with this photoaffinity labeling functional group, introduce it into the cell, and thus emerge with the photoaffinity labeled proteome, what seemed to us to be a major challenge was actually something to do with the downstream processing after isolation of the photoaffinity labeled peptides. And so what happens when you take this pool of enriched peptides that have been labeled by small molecules representing the binding site is that when you put this onto the mass spectrometer, all of this data analysis and database searching resulting in peptide identification is confounded and made more challenging when you have a small molecule that could have landed on any site in the proteome. So one way to look at this is by just kind of computationally characterizing how many amino acids could be labeled and what that size of the number of peptides in the database are present. And this is a log 10 scale axis. So we're drastically increasing our database size as a function of the number of amino acids that could have been labeled. This is just one of the major challenges. The other major challenge is the fact that the small molecule might not behave as we expect or desire on the mass spectrometer. And so one of the characterization by mass spectrometry that we need is to get enough fragments across the peptide backbone in order to characterize the peptide fully. So one of the challenges is in the case that we don't get enough of the fragments in order to site-specifically localize the small molecule. The small molecule might be metabolized, it might pop off the peptide, and it might internally cleave on its own. So for all of these reasons, it's very challenging to characterize the exact modification site of a small molecule, especially as that molecule gets larger. And so we wondered if we could come in with some kind of chemical validation mechanism to get around this. And so we were inspired by, in fact, work that I had done in Carolyn's lab to characterize strange glycopeptides to now use this similar strategy of isotope labeling and embedding a chemical identifier into the small molecule after it's enriched on the proteome. So our strategy is as such. So we take a cell, we introduce a small molecule with that photoaffinity labeled handle, label the small molecule and photoconjugate it directly onto the protein. We've now captured those proteins and we can enrich them with this chemical handle or this chemical tag using copper click chemistry followed by trypsinolysis to reveal the protein identification. And this is a typical chemical proteomics platform. However, we then take and cleave the biotin probe at this position. It's just a silane protecting group. So we have a mild acid hydrolysis. And by cleavage of this part of the molecule of the probe, which is now embedded into our conjugated peptides, we've effectively transferred this isotope label from our probe into only small molecule conjugated peptides. And we can then act differently and behave differently when we characterize these conjugated peptides by mass spectrometry. So one of the early work that we did as a proof of concept to show that this platform would work was towards the non-steroidal anti-inflammatory drug celecoxib. So celecoxib was initially developed as a selective inhibitor of cyclooxygenase 2 or PTGS2, selective inhibitor against PTGS1. And when inhibiting these proteins, it stops the second messenger arachidonic acid from being processed to prostaglandin H2, which then goes on to form additional molecules as further signaling progresses during the inflammatory process. So we were interested in using celecoxib to demonstrate whether or not we could identify broader targets of this molecule. And so we derivatized this toluene ring to the photoaffinity handle characterized by several biochemical assays that these molecules behave similarly, and then applied them in cells. So currently, one of our recent findings was that celecoxib actually binds and interacts with PGE synthase, a molecule downstream of cyclooxygenase 2. And this was encouraging to us because both of these proteins are actually interacting with the same small molecule prostaglandin H2. And the fact that these drugs, celecoxib and photocelecoxib, are also interacting was encouraging to see. Brief look at the data that we collected here. So we took photocelecoxib, introduced into various cell lines, including A549 cells, and identified the proteins by a typical chemical proteomics platform. And PTGES, that protein prostaglandin E synthase, was one of the top 3 proteins found in this list. We additionally then cleaved off our probe, so taking that photocelecoxib, enriching for proteins followed by tag cleavage, afforded the conjugated peptides, and now we're looking at direct data now of the isotope pattern that we've embedded onto the conjugated peptide, and then revealed by mass spectrometry using our directed searching method. And so here's an example of a mass spectrum that we discovered on PGES. And we followed up this work by showing that PGS is in fact stabilized by a thermal shift assay in the presence of celecoxib and photocelecoxib as well. So what we hope to use this kind of data for is that once we've discovered a binding site, we can take this information and embed it into the protein. And so fortunately, there's a crystal structure of this protein available that we were able to map the peptide onto. And we then wanted to essentially characterize where the small molecule is relative to this peptide. Because to us, being able to characterize exactly where the small molecule is liganding, we'll be able to most effectively use the kind of data that we're collecting for subsequent follow-up and both mechanism of action studies, but also the design of new ligands for different proteins. And so one of the major questions we wanted to ask here was whether or not we could more concretely define what these structural properties are representing. And so we took a two-pronged approach. One was we wanted to first develop an understanding of what different amino acids would be labeled here. So from another perspective, this is a crystal structure of celecoxib and the binding pocket with COX-2. And you have a series of amino acids that are equidistant to this particular point in that small molecule. And so if we change this point in the small molecule to a photoaffinity handle, what is the probability that we'd be able to hit these different amino acids? And so that's been the focus of one of our graduate students, Alexander West. It has been to essentially answer that very question. So if we have this diazurine equidistant to 2 different amino acids, and we photolyze it, what we would expect in a completely unbiased setting is a 50-50% ratio between the conjugation events. But realistically speaking, as chemists, and specifically as organic chemists, this seems like a very untrue situation. And so we wanted to fully elucidate what these yields would actually be. And so we dug through the literature, and there are a series of different diazurines that have been used throughout the history of diazurine chemistry, including this molecule here, which was actually synthesized by Fu, sitting next to me. And so we mainly looked at these diazurines, and they're primarily differing by the electronics at the alpha position. And we envisioned that that difference would actually impact how the reactivity would occur with different amino acids. And so we took this alkyl diazurine, one that's been quite popular in the field, both in our work and others, and we initiated our work here. And so when you take a diazurine and you photolyze it to generate a carbene, it actually, in fact, generates this carbene, but via a process of going through this linear diazo intermediate. The linear diazo intermediate then further photolyzes to this carbene, which can sit in a singlet or triplet state. Now, if this carbene is not close to a protein, say it's in water or loosely bound, what we would expect or hope is that that small molecule will get quenched. However, if that small molecule is tightly bound to a protein, what we would expect or hope is that the small molecule will conjugate directly to that protein. And the hidden secret with these alkyl diazurines is that about 50% of them just go to this 1,2 hydrogen shift or elimination pathway. What we found and what the data I'll show you points to a model of is that in fact this linear diazo sits in a conformation, it has this resonance structure, and that this resonance structure can react through an ion pair, creating a bias for different acidic amino acids found within the proteome. This alkyl diazurine reactivity was somewhat surprising, but essentially is analogous to a diazoesterification chemistry that you can perform in a reaction flask, just now being performed inside of a cell. So how did we determine that? The initial experiments that we performed were taking the taking these diazurine minimalist tags and incubating them in the presence of different amino acids and single amino acids that we were able to protect at the N and C terminus. We then incubated them in the presence of light, and upon that photolysis, we generate this linear diazo intermediate that then forms a carbene and inserts into a protein. So we initially did this completely neat under the model that this is the very closest that we can get a small molecule to a protein. And we wanted to evaluate what the yields were by NMR. So we added 4 equivalents of each of these amino acids, and each of these experiments were done in triplicate. We're looking at the amino acids that were able to react, although we've tested all of the amino acids. And indeed, we see some bias for different amino acids. This is not surprising. These are the more polar amino acids, what we would expect to be perhaps more reactive in this type of chemistry. However, when we add water to the solution, and now we added 1 millimolar of the amino acid, so it's a 1-to-1 equivalent between the diazurine and the amino acid, and we added water, we fully expected to compete off all of the cross-linking efficiency here. The idea being that water would quench the reactivity of the diazurine for the amino acid because of outcompeting it, being at 55 millimolar solutions. However, what we observed is not the case. We found that the 2 most acidic amino acids, aspartate and glutamate, are indeed reacting at a 15% yield. And if we increase the equivalence of the amino acids to 10-fold, this yield increases, and we see some reactivity for these next 2 amino acids, tyrosine and cysteine. So this led us to wonder whether or not we were actually seeing reactivity of this linear diazo species as compared to a carbene. And so we sought to slow down this transition. The data I've shown you so far was collected with a broadband wavelength over this full spectrum. And so we simply purchased a filtered bandpass filter in order to narrow out this wavelength and hopefully reduce this reactivity pathway. Indeed, by reducing that reactivity pathway, we see elevated yield, specifically for tyrosine and cysteine, for the diazurine, essentially showing a bias for these amino acids. And we think that the 2 acidic amino acids are already maxed out, irregardless of filtering out these wavelengths. This turns out to be completely pH dependent. So all of the data I showed on the previous slide was at pH was just in D2O, but now as a function of the titration of the pH, we can see that the yield is entirely pH dependent, implying that the hydrogen or deuterium that's coming from the acid is indeed coming from the acid. And we see that both with phosphate as a buffer, but also with glutamic acid are most reactive of the different amino acids. And this inflection point appears to be around the pKa of these molecules. So what this says to us is that these diazurines, specifically these alkyl diazurines, form the diazo intermediate that's quite reactive enough and biases us towards acidic residues in a pH sensitive manner. What we think is an interesting model is that as we electronically tune the alpha positions of these diazurines, both with fluoro groups as well as aryl groups, that this reactivity might decrease the reactivity of our intermediate diazo, giving it time to convert to the carbene and thus react with proteins in a more carbene-based manner as we expected. And indeed, the aryl trifluorodiazurine under similar neat conditions in the presence of individual amino acids, we now see reactivity across all of the 20 amino acids. We see a series of chemical We see differences in the types of products that are formed. In addition to heteroatom insertion, we also see CH insertion products. And most importantly, once you add water to the solution, even in the presence of 10 equivalents of amino acid, we don't see any reactivity at all, implying that the carbene-based mechanism is the primary mechanism that is being used here. So in addition to this study, we've also been characterizing very closely whether or not we can more concretely map the types of structural information that we can get out from this type of chemistry, from photoaffinity labeling chemistry. And here, Hope Flaxman, a graduate student in my group, and Chafu Chang, who's again sitting right next to me, headed up this project. So our idea here is that we would take photorapamycin and use it as a model system to characterize what the distances would be in terms of being able to measure the binding, in terms of being able to measure the binding affinity and binding interaction with a protein FKBP and FRB. And we specifically chose these two proteins, or chose this system, because it's been well characterized by others structurally, but also because it forms a ternary complex, and we're quite interested in that. So this project turned out to be quite involved, largely because rapamycin is a very large molecule and a large macrocycle, and it can form many of these unusual fragmentation pathways, including a first observation of a McLafferty rearrangement found by mass spectrometry. And so these are the two peptides that we were ultimately able to map. And fortunately, we were using our isotopic coding in order to identify these peptides confidently. When we map these two peptides back onto the crystal structure of rapamycin in the FKBP and FRB ternary complex, one of those peptides is on FKBP and the other is on FRB. We first took this information, and specifically the residues that were being labeled, and we performed a series of molecular dynamic simulations in order to characterize what is the minimum distance that this diazurine, specifically from the point on rapamycin, is able to label these different areas on the protein. And we came out with a 9 angstrom radius from this that we plan to use for future studies. In addition, a secondary finding from this work was that the diazurine and the cyclohexamide that this molecule is stuck on must be in 2 different conformations in order to label both of these proteins, implying that the diazurine chemistry and this photoaffinity labeling approach is enabling us to characterize these species in a dynamic manner. So all of this work has been kind of leading us up towards the work that I'm going to let Fu actually tell you about, is towards the singlepharins. And so this is work that I'm very excited to reveal to this audience today. Specifically, what we aimed to characterize was understanding further the structure function of singlepharin A and singlepharin B. So singlepharin is a natural product shown here. It was isolated from a Streptomyces species by Novartis back in the 1990s. And the way that they isolated this molecule was actually via its binding affinity to cyclophilin A protein that was well used and well loved in the immunosuppression field at the time. So they found that singlepharin A is a very tight binder for cyclophilin A. In fact, much, much tighter than what cyclophilin A is named after, which is cyclosporin. However, although singlepharin A is an immunosuppressant, it was subsequently found to have immunosuppressive activity that's via a novel mechanism. Structurally, singlepharin A is a unique macrocycle that has this 22-membered ring, which is the binding region shown here through this tripeptide for cyclophilin A. But on the other hand, we have this unique spirolactam functional group here. And it was this spirolactam that really intrigued us. Much of the mechanistic work prior to our work had shown that if you cleaved this singlepharin A across this olefin, you would arrive at 2 non-immunosuppressive fragments, the spiroazepital or the spirolactam and the cyclophilin binding molecule. While this maintained its activity for cyclophilin A, we were unable, or prior art was unable to really show whether or not and how the full activity of these molecules arose. Syngloferin B was also identified around at the same time from the same isolates. And the major difference is simply in this spirolactam, going from this secondary alcohol to this olefin. In fact, syngloferin B was more active than syngloferin A, and so we initiated our synthetic efforts towards syngloferin B. And so our ultimate goal was really from this perspective that cyclophilin A is binding to this part of syngloferin A, and that's PPIE here, and that there might be an unknown target that is at play here. While we were initiating this work, Greg Verdine and coworkers reported IMPDH2 as a potential target of PPIA mediated by syngloferin A, but of only PPIA, as there are several different homologs of the cyclophilins. And so we envisioned that coming in with a very systematic structure function study of the spirolactam would add a significant amount to this field. So now I'm going to let Fu tell the story about where we are with syngloferins, and go ahead.
So the strategy have been Christina and Leo Paquet have been made this compound. So that their strategy is steady coupling to assemble the stenin and the Markov side. So we adapted our strategy to this idea. And then we propose this more flexible synthetic strategy, basically spiraling co-structure that can suit this cyclization, this dihydropyrin, and this intermediate can be synthesized by these two assemblies, two fragment ketone and aldehyde. And those that vary the desired stereochemistries allow us to easy to derivatize on this each position. So the ketone, basically, there's a full stereochemistry here that can be established through this correlation. And this aldehyde, the two-state chemistry can install by using the Myers methodology. So you condensation the ketone and aldehydes treated with this sodium HMDS, and then you oxidize the secondary alcohol, then the acid immediate cyclization, you get this pyranol. This three-step functional manipulation, we get the 65% yield. And then now we need to cyclize. So first of all, we need to convert this benzyl ether to priamide. So through this three-step sequence, first of all, deprotection of benzyl group use red nickel and oxidize to carboxylic acid, and then convert to priamide when activation with carboxylic acid using the DSC. And then exposed to the ammonia. And now into this key intermediate, now we have to handle this cyclization. The idea is we want to convert this pyranol to the secondary alcohol, and then for instance to do the cyclization. So 1,2 reduction to get this pyranol to the secondary alcohol. And you can see here's this enone they can activate by Lewis acid. That depends on how you activate this compound. If you activate with the Lewis acid, and they can do cyclization and also elimination of secondary alcohol, and you will get the co-structure of a cyclophilin B. But if you not elimination, we just activate this position and then force the cyclization, and they will get the cyclophilin A. So this we play with a little bit chemistry here to let this compound become more powerful strategy. So here's demonstrate the cyclization to get cyclophilin B. So we treat it with one part with this trifluoroboroliterate, and we force the cyclization to get cyclophilin B. And the upper face, from the upper face, and then because this less stereo hinder, so that favor this this cyclization, we get single DR and very good yield, 77% yield. And then later we convert this, we had to make this stenin. So deprotection, we convert this PM EDT to stenin. Then, so first with deprotection and oxidize and treated with the here basement reagent, we're able to get the alkyne and good yield, 70% yield. And then deprotection this cyro ether, and then convert to the terminal stenin only E isomer, and the yield is 78%. So just as mentioned, we want to play with this cyclization. So we envision that you treat with different electrophilic reagent, you can basically derivatise on this position. For example, you put fluoride here, you can put the trifluoromethane, hydroxy group, even the methyl group, so that you can get the co-structure of synchrophenol A. So this idea, then currently we are working on this idea. And then once you have two fragments in hand, you're just coupling through this static coupling. We automate a little bit and improve the yield up to the 70% yield. And then final detection, this intramolecular tau, we have to get the finish this total synthesis synchrophenol B. Although yield a little bit lower, but we are working on that. With the similar, using similar strategy, we can coupling to get the code structure of synchrophenol A through this similar sequence, so we can finish this synchrophenol A. So now we have two compounds, and there is enough for us to further biological variation. So we want to use in the simple protocol, we just as mentioned. So this two position, we want to put the bifunctional photoaffinity tag, and then subject to the compound to to find target identification process. So these two compound, this synchrophenol put minimalist tag here and a metal tyrosine here, and then we subject to and also spiral tang alone, and then we subject to the target identification process. So spiral tang have been known to induce a homodimer. So they have a hypothesis that is really the homodimer is really important, is kind of important for the immunosuppressive activity, or actually that they just prevent the subprotein to engage. So we have two hypotheses here. So they either have subprotein through the homodimer complex, or it's just like, okay, you prevent, the homodimer prevent, but actually they have another protein that through this PPIA and synchrophenol complex. So we through this side exclusive column, we can find the synchrophenol A that can induce homodimer, but the compound we make not able to induce homodimerization. But you look at the cell availability, the compound 1, 2, and still kind of potent. So it look like the hypothesis more favor like homodimer is prevent the subprotein to engage. But so we more like favor this the PPIA synchrophenol complex to have the subprotein to come in. And then spiral tang alone, they not really have significant biological activity. So although this is very preliminary result, so currently still under investigation. So for this, okay. So so two compounds, synchrophenol A and 1 and 2 photo analog. We using our simple protocol, and these the one protein PPIA through this biological replicate the key appear in our mass spectrometry. There is PPIB, and then we also along the western block, and then that they can photo synchrophenol compound can enrich the PPIB and also compare off with the synchrophenol A. So although it's still very preliminary, but we will looking for the evidence to prove that this actually the real target for these two compounds.
Yeah. Thank you, Phil.
Yeah.
Okay. Yeah, so this is the current status of a project that we've been working on for a couple of years, and looking forward to feedback from this community about this work. So finally, I just wanted to sum up and that the work that we've been doing has been towards understanding how to regulate post-translational modifications, but also how to devise and understand how small molecules mediate protein-protein interactions in much the same way that PTMs can do. And finally, and most importantly, I'd just like to thank my group, specifically Chafu Chang, who's sitting right here, Hope Flaxman, and Alexander West, whose work I've told you about today. These sources of funding, and I guess thank all of you guys for listening. This has definitely been a very interesting webinar so far for us as well, to give a talk to ourselves. So thank you, Jeff, for organizing this, and I'm happy to take questions.
Oh, fantastic. There we have some virtual applause coming from all of our listeners. And we do have time for just a question or two. And we do have one in the chat room. So let's go ahead and start with that one from Jianu, who asks, can you introduce a cleavable linker between rapamycin and the diazarine so that you no longer involve the rapamycin in the peptide fragments?
Oh, hi, John. Thank you for your question. Yeah, we thought about doing this. So yeah, it's a great question. And essentially, could we introduce a cleavable linker here such that when it cleaves on a mass spectrometer or before going onto the mass spectrometer, we wouldn't have so much complexity. And so Peng Chen, I think and there's been recently another paper showing such a strategy using a selenium cleavable linker to mark different proteins. And we've certainly thought about it. In fact, Fu here has thought about it a bit more than I have because he's developed some small molecule tags towards it, but they're still very much in development. So happy to talk with you about it more too.
So one other quick question is for those of us who don't do this kind of mass spec analysis very often where you're looking for pattern matching. How do you do that? Could you walk us through the workflow of how that actually works?
Sure. Let me see if, yeah, so this is actually a great slide for that. So essentially what we're doing with the pattern matching is we're essentially comparing the isotopic distribution of this distribution here, which is a patterned species, to the isotopic distribution of a unpatterned species. So if you essentially subtract out these two first lines here, that would be the equivalent of an unpatterned species. And so essentially by comparing those two to each other, we can take the real data here that was observed and compare them to the predicted data. And by comparing it to the predicted data, we can determine that this species here is more like a pattern species and therefore more likely to be carrying a small molecule on the peptide.
Okay. Okay, well, thank you again for your talk, and good luck with everything moving forward. It looks like an exciting project, and we look forward to hearing more about it in the literature and maybe venues like this in the future. So at this point, I'd like to go ahead and introduce our second speaker, which is Carolyn Bertozzi. Professor Bertozzi is the Ann T. and Robert Bass Professor of Chemistry and Professor of Chemical and Systems Biology at Stanford University, where she moved fairly recently. She received undergraduate degree in chemistry from Harvard, a PhD in chemistry from Berkeley, and then did a postdoc at UCSF in the field of cellular immunology. She was at Berkeley, as I mentioned, for many years, and then now a couple of years ago moved to Stanford University, and has been involved heavily with Stanford's Chem H Institute at that institution. Of course, Professor Bertozzi has many honors and recognitions, but I want to just mention two relevant to this audience. One is that she is a founding editor of ACS Central Science, and in that role plays a great deal of, does a great deal of important service for the chemical community. And of course, a few years ago, she was the COPE awardee, which is one of the highest divisions awards of the Division of Organic Chemistry. So without further ado, I'm going to ask if we can transfer the screen over to Professor Bertozzi so she can give her talk.
Okay, let's see. Hi, I'm trying to share my screen, but it doesn't seem like it's happening. Can you guys hear me?
We can hear you, but we just see your name. That's what I see in my screen.
When I say share and I click on share, it just bounces back to desktop.
Okay. Do you have a box for sharing PowerPoint, perhaps?
You're going to need to turn your video back on.
Oh, got it. Okay, thank you.
Perfect.
All right.
And I think that should let you share your screen.
How's that?
Yep.
Thank you. Okay. So first of all, Jeff, thank you for the introduction and for inviting me to participate in the symposium. And it's an extra special honor for me to follow up from Christina's beautiful talk, and a great opportunity for me to kind of catch up with all the great stuff she's been doing. And I thought what I would do is talk about a fairly new project in our lab around the development of a new therapeutic modality that we call the LITACs. And that's an acronym for lysosome targeting chimeras. This is a project that now I'm trying to advance my slides and nothing is happening. Any suggestions, folks, on how I can get my slides to advance? All right, I'm going to exit PowerPoint.
Click on the slide.
The microphone now is on.
Oh, okay, I got it. It's working. All right, so the work was initiated by postdoc Steve Bannock, who I contemplated inviting to join me this morning, but he's in the throes of submitting job applications for academic positions, and it's a very big week with deadlines. So I excused him, and I'm standing up in his place. And the supporting cast are graduate students Kayvon Pedram and Green On, as well as postdoctoral fellows Nick Riley and Simon Wisnowski. So the backstory is that there's a lot of excitement around a new modality for small molecule therapeutics, which are targeted degraders. Sometimes people refer to them as proteolysis targeting chimeras, or PROTACs for short. And it was inspired by that acronym that we created the LITAC acronym. But how these small molecules work is that they have two moieties. They have one element that targets a protein of interest that one might want to degrade, and it's a ligand that can bind that protein on any site, and it doesn't have to be an active or functional site. And then the other part of that molecule binds to an E3 ligase enzyme. And these are enzymes that initiate the process of ubiquitination, wherein that target protein that's in proximity by virtue of binding to the PROTAC molecule acquires ubiquitin chains that mark it for degradation by the proteasome. And there's a lot of activity in the development of these so-called PROTACs these days because of the promise that they could expand what we call the druggable proteome. And that's because many target proteins of interest don't have a small molecule drug binding site that also blocks their function, and therefore they may be considered undruggable. But being able to ligate them in any crevice that's convenient might then make them druggable by this degradation strategy. And in addition, some people think there are benefits to degrading a target rather than just blocking it with a small molecule ligand. And that's because when you degrade the target, you've just completely taken it out of the cell, and your molecule has the potential to be catalytic rather than stoichiometric. And just to give this audience, which I realize is an organic chemistry audience, a view for what such a PROTAC might look like, here's a molecule in which this was developed at a company called Arvinus, which was founded by Craig Cruz. And he was really the first person to sort of coin the term PROTAC and to reduce it to practice in the laboratory almost 20 years ago now at Yale. But you can see this is a molecule that is bifunctional. The part on the left binds a target of interest that happens to be involved in epigenetic modifications. And the part on the right is an analog of thalidomide, which binds the E3 ubiquitin ligase cereblon. And then there's a spacer in between. So this is a wonderful strategy that now has really taken off in the pharma industry. but it's not without some limitations. Most significantly, the targets that you can go after with the conventional PROTAC, they have to be cytosolic or at least have a ligandable domain that faces the cell cytosol, because that's the location of the proteasome and the E3 ligases. And there are a lot of interesting targets one can go after in the cytosol, but almost half the proteome is not in that compartment. So about 40% of human proteins are predicted to be either secreted into the extracellular environment or associated with the plasma membrane and lacking a cytosolic domain that is amenable to small molecule ligand development. And it was this part of the proteome that we thought about targeting with the LITACs. So the LITACs are going after another major pathway of degradation for cellular proteins, and it's the endosome-lysosome system. So this is the pathway by which most extracellular or cell surface associated proteins get degraded. And it starts with internalization of those targets through a variety of endocytic pathways that bring those targets from outside the cell into intracellular organelles called early endosomes. And those early endosomes go through a maturation process to eventually become late endosomes. And throughout that process, there is a gradual acidification of the internal part of those organelles. And then eventually, a lysosome-endosome fusion event ends up delivering the cargo to lysosomes, which are organelles that have degradative enzymes like proteases, glycosidases, hydrolases, and also various redox and oxidative enzymes. So that's kind of a garbage disposal for those extracellular and cell surface proteins. So we thought maybe we could develop bifunctional molecules that take hold of those extracellular proteins and somehow bring them through this pathway and deliver them to the lysosome for degradation. So to do that, we took advantage of a well-known trafficking system, which is naturally used by cells to basically deliver those degradative enzymes that the lysosome needs to do its digestive functions into that organelle. And it's a pathway called the mannose-6-phosphate receptor pathway, or the M6PR pathway. So the way this works is normally your lysosomal enzymes are initially biosynthesized in the secretory pathway. So that's the ER and the Golgi, which is these little cartoons. And within the Golgi compartment lies this mannose-6-phosphate receptor, and those lysosomal enzymes get glycosylated in the secretory pathway, and they get modified with this mannose-6-phosphate mark. It's quite a unique element to see on protein-associated glycans, and it's quite restricted to these lysosomal enzymes. So that's how the receptor finds them in the Golgi and then delivers them through a trafficking pathway that is not actually all that well understood, directly from the Golgi to late endosomes. And from there, where it's a relatively acidic environment, the mannose-6-phosphate receptor lets go. It has reduced affinity at that pH for mannose-6-phosphate. And those enzymes can then progress and find their way to the lysosome, and they function in that compartment. What's interesting is that the mannose-6-phosphate receptor, in addition to trafficking between the Golgi and these late endosomes, it also cycles to the plasma membrane. So at any given time, if you took a snapshot of the cell, you'd find about 10% of that receptor is exposed on the plasma membrane, where it is available to grab mannose-6-phosphate modified proteins from the extracellular space, and it then brings them through an endosomal pathway through clathrin-coated PIT-mediated endocytosis to the early endosome and then to the late endosome through this other kind of backdoor. And from there, those extracellular modified enzymes can then find their way to the lysosome. And what's interesting is we don't actually have a good understanding of why cells might do this, why they might want to grab mannose-6-phosphate modified lysosomal enzymes from outside the cell. Because for the most part, we think of our cells synthesizing their own lysosomal enzymes and directly delivering them to the endosome. But it is a convenience that cells can do this because this is how enzyme replacement therapies work, the ones that are used to treat lysosomal storage disorders. So, companies like BioMarin and Sanofi Genzyme make recombinant lysosomal enzymes for patients with rare genetic diseases where the genes encoded those enzymes are damaged. And all one has to do is infuse the recombinant mannose-6-phosphorylated enzymes into the patient's circulation, and cells just naturally take them up through this pathway and deliver them to lysosomes. So we knew that recombinant proteins could be infused into patients and they just find their way to lysosomes. And so the question was, could we hijack this pathway to target unwanted proteins, like disease-related proteins, and get them delivered to lysosomes and degraded therein? So what Steve did is he developed bifunctional LITAC molecules that have two parts, a part that binds the target and a part that engages the mannose-6-phosphate receptor. And for most of our work, the target binder is an antibody, a monoclonal antibody molecule, but one can also use small molecule ligands or really just anything that binds with an appropriate, ideally high affinity. And then for the mannose-6-phosphate receptor engager, we made oligomers that were glycopeptide based with mannose-6-phosphate residues. And the reason we used oligomers is that there was some literature suggesting that the mannose-6-phosphate receptor needs to be clustered in order to induce internalization and trafficking from the plasma membrane to those late endosomes. So the way that we would use these LITACs would be to target either soluble proteins floating in solution, and that would be like this purple box floating outside the cell. And we call this a target that you would want to degrade in trans, meaning the LITAC molecule would bind to it separately from the cell, and then the mannose-6-phosphate receptor on the cell surface would snatch it out of circulation and bring it into the lysosome for degradation. But we also envisioned using LITACs for targets that we call in cis, and those are targets that are on the plasma membrane. So the binder could interact with those targets, and then on the same cell, the mannose-6-phosphate receptor would engage these ligands and induce internalization. And so whereas the situation where the target is in trans was kind of precedented through the use of these enzyme replacement therapies, there really wasn't any known natural biology where the mannose-6-phosphate receptor would engage a target and degrade it in cis, or internalize it at least in cis. And so that was kind of the big open question in this project. So for the organic chemists, the way that we synthesized the receptor engagers was polymerizing N-carboxy anhydride functionalized glycosylated amino acids. So here's an example of a monomer that we used in a NCA polymerization reaction. And this is the nickel catalyzed NCA living polymerization chemistry that was developed at Tim Deming's group at UCLA. And up here is the mannose-6-phosphate group, and it's protected with acetyl esters on the hydroxyl group. And this is the dibenzyl phosphate diester as a protecting group. So this chemistry, followed by deprotection, allowed us to assemble oligomers of various lengths, where we had basically blended together in these polymers serine-modified mannose-6-phosphates, as well as alanine residues, just to kind of space them out a little bit. And in a proof of concept experiment, what Steve did is he took those oligomers and then he functionalized one end of those polymers simply with a biotin group. And the idea was to see whether this synthetic molecule could grab a protein in solution, and that was a fluorescent dye labeled neutravidin protein in this experiment, and take it up and deliver it to the lysosomes. And we put since there's a fluorescence tag on the neutravidin, we could simply quantitate uptake into these cells using a flow cytometer. So here are some data showing the fluorescence uptake of cells. And this was an experiment that was done with, I think it was K562 cells. It's down at the bottom there. No, it's not. Yeah, these are K562 cells. And what he looked at was the uptake of LITAC molecules that had different sugar modifications. So one of the analogs had the mannose-6-phosphate groups, and he looked at both short oligomers that were about 20 mers, as well as longer oligomers that were around 60 mers. But we also looked at an isoster of mannose-6-phosphate, which was mannose-6-phosphonate. And that was because there was literature from the enzyme replacement therapy field showing that one of the liabilities of mannose-6-phosphate on natural enzymes is that it can be cleaved by phosphatases in circulation. And that limits its uptake by the lysosomal trafficking pathway because the free mannose residue that's released after dephosphorylation is not recognized by that receptor. And even worse, those free mannose residues are recognized by an uptake receptor on circulating macrophages, which then ends up being basically a mechanism to sequester these molecules. And all of that can be avoided by simply making this phosphonate substitution, because now it's non-hydrolyzable and therefore it cannot be degraded in circulation. And in addition to those two, Steve also made control Litacs with different sugars like free mannose and acetylgalactosamine. And what you can see from this bar chart is that whereas the control sugars that don't recognize the mannose 6-phosphate receptors were not taken up above a background level by these cells, any Litac molecule that had mannose 6-phosphate, either short oligomers or longer oligomers, or mannose 6-phosphonate, either short or long oligomers, these were all taken up by the cells. And that uptake was inhibited by having an excess amount of free mannose 6-phosphate in solution, which competes for binding to the mannose 6-phosphate receptor. Okay. Once we had this assay up and running, we realized that we could use this uptake phenomenon as a means to do a CRISPR screen to try and get a better understanding of the actual mechanisms of mannose 6-phosphate receptor cycling, which were not well understood. It's interesting because the pathway itself was discovered back in the 1970s, and this was sort of classic glycobiology 101 work that had been performed by Stuart Kornfeld's group at WashU St. Louis. And it had gone into the textbooks, so you could read about it in the 70s and 80s. But here we are in the modern era with all of these great genome-wide mechanistic tools that we can apply to actually understand the details of what's involved in the cycling. So we did that. We took a K562 cell line that had been transduced with a single guide RNA library that basically covers all of the open reading frames in the genome. So this is what we call a CRISPR-I pool. So every cell in this pool ideally has one gene that's been knocked down. And then what we did is we sorted the pool with a flow sorter to identify those variants that had lost the ability to take up our Litac molecule bound to the fluorescent neutravidin. So the cells with low fluorescence were sorted out. And then we did next generation sequencing to figure out which guides had been enriched in that pool that were no longer taking up the Litacs. And from there, we were gratified to see that one of the strongest hits was the gene encoding the mannose 6-phosphate receptor, which is this blue dot right here. But as well, we found many other genes that were enriched in that pool. And what was interesting is that we found many genes from what's called the exocyst complex. And that is a complex of proteins that were known to be involved in vesicle trafficking processes for other proteins in the cell, but we now know that they also contribute to the recycling properties of the mannose 6-phosphate receptor. And when we took all of the guides that were enriched in the screen and sort of mapped them onto what are sort of known or putative functions for those proteins, all of them mapped into these various categories that made good sense for this pathway, such as genes that encode proteins involved in lysosomal membrane biology, vacuole and coated vesicle biology, processes having to do with Golgi transport, and so on and so forth. So we're actually learning something about the fundamental biology of mannose 6-phosphate receptor cycling with these Litacs. Okay, so having done all that, we then moved to some more interesting Litacs against targets that had therapeutic relevance. And for this, we made Litacs where the binding element was a monoclonal antibody. So we assembled these just using some pretty straightforward kind of old school bioconjugation chemistry. So we called these our first generation quick and dirty Litacs, although I wouldn't say that if Steve were sitting here next to me, but this was sort of the easiest way to put them together, which is that we made the glycopolypeptides using that polymerization chemistry where there was a free amino group at their N terminus. And then this allowed us to do some non-specific conjugation to antibodies that we had already functionalized on their lysine residues with little linkers with azide groups. So these free amino terminal glycopolypeptides could be conjugated to an NHS ester cyclooctine reagent, and then we just click together these two to attach the Litac to the antibody. And again, it's kind of heterogeneous because these azido linkers were kind of sprinkled throughout lysine residues on the surface of the antibodies, and that's nonspecific. And as well, the glycopolymers, even though the NCA chemistry, the nickel catalyzed chemistry is a living polymerization with fairly low polydispersity indices, it's not like 1.0. So there's some heterogeneity in the polymers as well. And you can kind of see this when you analyze these molecules on native gel electrophoresis. So an unmodified monoclonal antibody on a native gel has this typical banding pattern where these different bands represent a combination of different glycoforms, as well as probably also some various proteoforms. And once you do the chemistry to sprinkle basically these linkers onto the lysine residues, the mobility increases in the native gel because you lose positive charges when you modify or acylate the lysines. So the protein has a higher net negative charge, so it migrates a little faster. But it migrates even faster if you now conjugate on these mannose-6-phosphate glycopolypeptides, because those are polyanions, right? So they really drive the migratory potential in the native gel. And the first antibody we used was an antibody called cetuximab. And the reason is that, oh, whoops, sorry, my slides are a little bit out of order here. Sorry, skip that. That cetuximab binds a protein called the epidermal growth factor receptor. And it is a protein that is upregulated in a number of solid tumor settings. And cetuximab is a clinically approved, it's a medicine that's used clinically to treat certain solid tumors that have the EGFR expansion. So this is an approved drug. So what we did is we made a LITAC out of cetuximab and then asked the question, can this antibody not only bind to EGFR, which is its normal activity, but also drive the internalization and ultimately the degradation of EGFR. And initially we could see a pretty dramatic effect just by simply doing fluorescence microscopy on HeLa cells, which are cervical cancer cell line that overexpressed EGFR. So you can see, this is the image on the left. These cells are stained both with a reagent that lights up the nucleus, that's the blue fluorescence, that's DAPI, as well as with an antibody that binds EGFR and has a fluorescent dye on it, and that's the pink fluorescence. And this is an antibody that we use for staining, which binds in a different place than cetuximab. So it's not affected by cetuximab binding. And you can see that naturally on the HeLa cell, EGFR is highly localized on the plasma membrane. And when you treat those cells with the naked antibody, there's no change in the distribution of EGFR. But when you treat those cells with the LITAC version of cetuximab, EGFR has been driven into the cell and it's concentrated in the lysosomes, which we've shown independently with lysosomal markers. More importantly, this internalization leads to degradation. So these are Western blots that show the steady state levels of EGFR protein on HeLa cells under different conditions. And vinculin is just a loading control to show that the same amount of protein or total protein was administered to the gel in all of these experiments. So when you just break open HeLa cells and look at how much EGFR there is, you can see this is the intensity of the band on the Western. And if you treat them with the ligand for that receptor, which is the epidermal growth factor, that's known to bind EGFR and drive its internalization and also its degradation. And that's exactly what we saw. So EGF treatment leads to reduction in EGFR levels. And if you treat the cells with the naked antibody, cetuximab, which is known to bind but not induce internalization of EGFR, you can see there's essentially, no significant change in total protein level. But when the cells are treated with the cetuximab LITAC, there's a dramatic 80% reduction in the steady state level of that protein. And that's exactly what we were hoping to see with the LITAC. By contrast, if we did the experiment in HeLa cells that had been transduced with dCas9, as well as a single guide RNA that is against the mannose-6-phosphate receptor, now that reduction in protein seen on the left blot is lost on the right blot. So that tells us that the degradation induced by the LITAC is entirely dependent on expression of the mannose-6-phosphate receptor, as we had hoped. Okay, and just to show you what the kinetics of degradation look like, on day one, if we treat these HeLa cells with nanomolar concentrations of this LITAC against EGFR, you can see there's a progressive degradation that sort of reached the steady state around 20% out after many days. And it's a pretty durable effect. And when we kind of did some math on the back of the envelope, we convinced ourselves that the cetuximab LITAC was actually catalytic in that one molecule was able to turn over and degrade more than one EGFR target. Okay. We also did some quantitative proteomics to see what the kind of specificity of the LITAC was. And so these are volcano plots where basically you can quantitate the level of protein, and anything that changes between treatment with cetuximab alone or with the cetuximab LITAC would show up as a dot that's either going down, which is in the blue upper square on the left, or if it's going up, it would be in the orange upper square. And what we found is that whereas EGFR doesn't change at all levels when you treat with cetuximab, treatment with the cetuximab LITAC caused a significant downregulation of that protein's abundance. And what's interesting is there are a few other proteins that significantly changed in their abundance as well. And we believe that these are proteins that could be in association with EGFR and therefore kind of indirectly degraded because they're in a complex, or they might be dependent on EGFR through downstream signaling events. And that also can teach you something about the interactome using this technology. Okay. We couldn't help but think about PD-L1 as an interesting target. As many listeners might know, PD-L1 is the target of Genentech's antibody called Tecentriq. And Tecentriq is a cancer immune therapy, and PD-L1 is the ligand for PD-1, which is the T-cell checkpoint receptor. So Tecentriq falls into the category of checkpoint inhibitor therapy, and this is a very hot area in oncology. PD-L1 also has a very interesting recycling biology of its own. It sits on the plasma membrane, but it has a cytosolic domain that interacts with a chaperone called CMTM6, which is the green squiggle. And what that chaperone allows is that PD-L1 can continuously recycle from the plasma membrane through a recycling endosome and then back to the plasma membrane again. And so when CMTM6 is expressed, it allows elevated levels of PD-L1 on the plasma membrane. And if you knock out CMTM6, there's a redistribution of PD-L1 so that there's less of it on the membrane, more of it gets internalized and degraded. So the CMTM6 protein is kind of like a dial that can actually dial the expression level of PD-L1, which is relevant to cancer immune suppression. And we were curious if we made a LITAC against PD-L1, which would then drive it through the mannose-6-phosphate receptor internalization pathway, how would that compete with the natural CMTM6 mediated recycling pathway? It's kind of like a tug of war of different internalization mechanisms. And what we found is that a LITAC is able to enforce degradation of PD-L1, at least to some extent, even though it's got to compete with another recycling mechanism. And that's shown here. So when you treat cells with 25 nanomolar of various reagents, including the Tecentriq mannose-6-phosphate LITAC molecule, that molecule induces degradation so that you reset the total protein level down around 70% lower than when you started. And if you do the same experiment with excess free soluble mannose-6-phosphate that competes with the receptor, you can reverse that degradation, showing that it is receptor specific. Okay. Finally, I want to point out an interesting feature of the system, which is that we can tune the affinity of the mannose-6-phosphate receptor engager by basically changing the phosphate to other anionic isosteres. So it was known from the literature on this receptor that mannose-6-phosphate and phosphonate bind with similar affinity, but if you change that anionic group to a carboxylate, either this alpha-beta unsaturated carboxylate or the saturated analog, you can reduce the affinity of receptor binding by several fold. And we thought this could be useful because in the situation where you're targeting a cell surface molecule, which is, again, we call that in cis, you might want to have a lower affinity receptor engager, one that won't bind significantly in trans, but can still bind in cis when the effective molarity of the receptor is higher because you're on the same membrane. And what we showed is that if we make a LITAC, so here what we did is we made a cetuximab LITAC, so it binds EGFR, using mannose-6 carboxylates instead of mannose-6 phosphonates. And what we found is that if you take that LITAC and just incubate it with cells just that don't have EGFR, for example, Jurkat cells or a T-cell lymphoma, and they don't express EGFR. By flow cytometry, there's no significant binding of the LITAC, which is this blob of cells on the right, compared to when you just treat with cetuximab alone. So the low affinity mannose-6-phosphate receptor ligands are not good enough to have binding in trans. But when we looked at a degradation experiment, the LITAC against EGFR made with mannose-6 carboxylates was every bit as effective in driving degradation as was the phosphonate version. And that's seen here. So EGFR levels on HeLa cells without treatment. Here's EGFR levels after EGF exposure, so ligand-mediated internalization and degradation. And then finally, cetuximab alone no effect. And here's cetuximab mannose-6-phosphate LITAC and cetuximab mannose-6-carboxylate LITAC. So low affinity ligands, perfectly effective to drive degradation in cis, but not so much in trans. And you might want that in a therapeutic setting where you want the LITAC molecule to find its way to the tissue of interest and not just get taken up in trans by every cell in the body, since every cell expresses the mannose-6-phosphate receptor. Okay, so with that, let me give you a little view to the future. What I showed here was just the kind of first generation LITACs where we had non-specifically conjugated heterogeneous polymers around the surface of an antibody. And those were easy to put together quickly, but they're not what you might want to have a therapeutic reagent. You want something that's, I think, more refined, more chemically defined, and more optimizable. What I didn't show is we had also made some even quicker and dirtier LITACs where we had stitched mannose-6-phosphonate groups through isothiocyanate chemistry directly onto the antibody surface without even making oligomers. And those are easy to make and they can have LITAC activity, but again, for therapeutic development, I think we need something more refined. So right now, Green On, who's the grad student I mentioned up front, is making LITACs that have more chemical definition, where we attach the mannose-6-phosphate receptor engager onto antibodies with site-specific chemistry. There are a number of ways to do that, including one that we had developed in our lab. We have also stopped using heterogeneous polymers, and we now make synthetic glycopeptides of homogeneous length. And we've learned that they don't have to be very long at all. They can just be between 2 and 5 amino acid residues and have perfectly good LITAC activity. And we're also interested in architectures where the mannose-6-phosphonate groups are displayed not in linear arrays on a peptide backbone, but in branched arrays on dendrimeric scaffolds. Sorry, what happened? Okay, I just clicked something and I don't know, what are you guys seeing my slides still? I had a pop-up window.
Yes.
Okay, sorry. All right, so, and also what Green is doing some structure activity correlation studies to figure out what is the best geometry for a LITAC, especially when the binder is this big antibody molecule. Because, you know, in principle, the antibody binds the target and it's just a spacer between that target and the mannose-6-phosphate receptor. And we assume there's probably some optimal geometries. The good news is because we do have a way to systematically move site-specific modification sites around the antibody surface, we can make LITACs with these different display. So Green has coined this acronym PAR, which is the peptide antibody ratio. It's kind of a takeoff on people from the antibody drug conjugate world would call this a DAR, the drug antibody ratio, but this is the PAR. And she's now making constructs where we dangle the mannose-6-phosphate receptor ligands from different places like the hinge region, the C-terminus, off the light chain, off the heavy chain, and where we have not just 2 copies of the receptor binders, but maybe 4 copies in different geometries. So we'll be able to line these up head to head and try and figure out what is the most effective LITAC structure that you might want to progress into a therapeutic model. Okay, so with that, let me just leave you with kind of a view of the breadth that we think this modality could have for making new kinds of therapeutics. And there's a number of applications that we're pursuing in my lab, and we've also had people reach out to us with their ideas for collaborative work. And these include cell surface molecules that are otherwise, you know, kind of undruggable, you know, proteins that are not receptors, or they're not enzymes, they're not that amenable to small molecule ligation. Maybe you can generate antibodies against them, but the antibodies are not function blocking. And so we have some ideas in that space. Of course, there's lots of interesting molecules that already have antibodies or small molecule ligands that might be effective for a while in the therapeutic context, but against which resistance often arises. And we think maybe the LITAC might be an alternative strategy for those kinds of targets. Targets that really need to be internalized to be effective, and antibody drug conjugates are a great example of that. The LITAC approach drives internalization, and so fusing that with an antibody drug conjugate architecture might be an interesting way to potentiate those molecules. And then finally, there's all kinds of circulating molecules that are just pathogenic and you want to get rid of them, and And these include immune complexes and amyloid conditions. And so we're looking into applications like those as well. And with that, I'll just thank once again, I mentioned the main contributors right up front. The driver of this project is postdoc Steve Bannock, who, like I said, is on the job market. So if you're on a search committee, keep your eyes out for him. But he is supported by a wonderful team of people, all of whom have contributed in various ways to this work. So thank you, and I can take some questions.
Thank you very much. As we wait for the virtual applause to die down, there's one question that came up fairly early in the talk, and I think you might have addressed it a little bit at the end, but it pertains to the choice of polyalanine or partial polyalanine as a linker and what characterization you do of linkers. Do you want to expand a little bit more on that?
Sure. Let me go backwards. Oh, how do I go backwards? Oh, there we go. Okay. Let me just get a view to what these things look like. So, so I think the question is, these glycopolymers, how do we characterize them? And why did we sprinkle alanines in there? And I have the unsatisfying answer is that we were already doing that. We were already making glycopolymers of those types for other projects in the lab. So we just kind of used that approach because we were doing it. We kind of knew how to do it. And why in earlier work we were making these serine glycosylated alanine sprinkled polymers is because they emulate a class of glycoproteins called mucins. And we have a longstanding interest in the biology of mucins. Now, mucins don't have mannose-6-phosphates on them. Mucins have GalNAc and then elaborated glycans. And we had just been making them for lots of other projects in the lab. So when Steve, wanted a quick way to test this idea of putting mannose-6-phosphate oligomers onto an antibody or a binder, he just used this. We characterize them using all the typical polymer characterization techniques. We do NMR, we do light scattering analyses, gel permeation chromatography. So nothing out of the ordinary for polypeptide analyses that are polypeptides made by NCA polymerization. Does that address the question?
It did, as far as I could tell.
Okay.
So I guess a more basic question is that all of the linker designs that you're using are using multiple copies of M6P.
That's right.
And from a very basic perspective, what is the reason for that?
So again, there was literature suggesting that a one-to-one binding interaction between a cargo and the mannose-6-phosphate receptor is not enough to induce effective uptake, and that clustering of the mannose-6-phosphate receptor drives an internalization program. So we wanted to make sure that we could cluster. We made these polymers early on, these 20 mers and 60 mers, because we thought those are big enough where they could grab multiple receptors in the vicinity and pull them together. Since then, what Green has found, I'm going to go back to that later slide. Okay, what Green has found more recently is that putting 20 mers and 60 mer glycopolypeptides in multiple copies on these antibodies, it was way overkill. What we now realize is you really, she's made oligomers where it was only 2 mannose-6-phosphate groups. And if you have 2 mannose-6-phosphate groups on a oligomer, on a little peptide, and then you sprinkle 2 of those on an antibody, that seems to be enough. So I don't think you need huge clusters of mannose-6-phosphate receptors to drive internalization. You might just need a few. But we haven't really worked it all the details out. I think being able to do this will help a lot, because if we can make the molecules where we know exactly where the mannose-6-phosphate groups are positioned, the distance between them, their valency, and systematically vary that, I think we can find what's the optimal structure. We don't know that yet.
I'll mention that the person who asked the first question has responded with thank you. So obviously you did answer that to satisfaction. And then the last question would be, you mentioned in passing that maybe you might not need to use antibodies and that small ligands might be okay as well. Have you tried that? Have you have any sense of what the parameters might be in terms of how tight a binder you would really need for this to fly with a non-antibody bait?
That's a great question. So the I mean, I guess you could say that the biotin streptavidin was kind of a small molecule LITAC, right? Biotin is a small molecule ligand that we put on the mannose-6-phosphate oligomers to grab a soluble protein. We have not yet tested cell surface protein LITACs with small molecules, although that's a project in the works. And I think it would be nice to have a system where we could systematically vary the affinity of the small molecule ligand for the target protein to ask exactly these kinds of quantitative questions. So we don't have an answer for cell surface molecules, whether a small molecule LITAC is effective, but I guess in principle, if you can just get the right geometry to engage both the target and the mannose-6-phosphate receptor, they should be, and we'll see.
Okay, I think that pretty much brings us to the appointed hour here. I'd like to thank both Professors Bertozzi and Wu for outstanding talks and for participating in our ongoing series. Invite everybody to keep in tune for when we do the next one. And thanks again to folks at Emory, Rachel Hall, Carl Hansen, Steve Silverman, our folks at DOC who make this happen. So thanks all, and a good day to everybody.
Thank you.
Cheers. Bye-bye.